Okay, I'm just going to give a couple of minutes now on a few leftovers that we have from the trafficking and assembly session. All right, one of the things that we haven't talked about is how nucleic acid is recognized and how is it packaged. It can be efficient and very specific. So if you think about it, retroviral genomic RNA constitutes only about 1% of an infected cell cytoplasmic RNA, and it looks like cellular messenger RNAs. It's capped, it's polyadenylated, but it is the RNA that's packaged in most retrovirus particles. So how does that happen? We'll talk about that. It poses a specific problem for segmented viruses. So, for example, influenza virus or reovirus. So for influenza virus, it's possible that packaging the segments is random. So perhaps there's a signal that makes it clear that these are viral RNAs, but how do you get one each of the 8 genome segments into an influenza particle? Or how do you get 12 orbivirus genome segments or 10 reovirus gene segments? And so people have done studies for influenza viruses for a long time. And the idea is that if maybe it's not selective, maybe we don't know what makes a HA segment different than a neuraminidase segment different than a nucleoprotein segment. If packaging of the influenza genome segments was random, that would probably yield one infectious virion out of every 400, and actually that's completely within the particle to PFU ratio of influenza virus. If, on the other hand, you packaged more than 8, if, say, there was enough room in an influenza virus particle to package 12 segments, 10% of those would contain a complete genome. So there are different ways to think about how this might work if packaging the different segments was random. But actually, there's some new evidence, fairly recent, that there are specific sequences on the different RNA segments of influenza virus so that they are selectively incorporated into influenza virus virions. And this is enabled now because there are mechanisms, there are methodologies to mutate cDNA copies of the different genome segments and reconstitute an infection completely out of genetically engineered RNAs. So that's a particularly challenging question, and the jury is still out for segmented viruses. You can imagine, just one last point on that, you can imagine that for an enveloped virus with helical nucleocapsids, those are not, those have a sort of pleomorphic structure. You could conceivably package more than 8 because the particle would just get bigger. That tends to be more challenging to think about for the non-enveloped viruses, the icosahedral viruses, like reovirus that are segmented, because there's a fixed volume, right? The icosahedron, there's a limit as to how much genetic information you could cram in there. So if you wanted to put more than 10 segments in, that might be problematic. Okay, so how do we understand this? How do we study this experimentally? We know for a number of viruses that discrimination of what is genome and what needs to be packaged and what is not is the result of unique sequences or structures called packaging signals that are on the viral genome. And on this slide, from figure 13.12, packaging signals are illustrated with these little arrows up in the adenovirus segment up on the top, which is just part of the genome. And here down on the bottom, you see SV40. And here in the, actually close to the origin of DNA replication, close to the early transcription unit in the transcriptional control region, overlapping with these SP1 transcription factor binding site is the packaging sequences. These are defined experimentally, so how do you do that? If you mutate them in clones, that genome is no longer packaged into the virion, so you muck it up and you get a negative outcome. And then importantly, if you add those sequences to a foreign piece of nucleic acid, that sequence or structure directs the encapsulation into the virion of that foreign nucleic acid. So both things are actually important. And as I just said before, there are packaging limits. There's an upper limit on the amount of viral nucleic acid that can be accommodated into an icosahedral capsid, usually no more than 5 to 10% bigger than the viral genome. And that's important when you start to think about how people are using some of these viruses to deliver genetic information, novel genetic information for gene therapy purposes. Okay, let's look at what we understand about this in retroviruses. And so for retroviruses, the packaging signal has been called psi, PSI, the Greek letter here. And in Maloney-Murine leukemia virus, which is down here in panel B, you can see that the psi sequence is downstream of the 5' splice site, right? So remember that for retroviruses, there is one primary transcript, and that for these viruses, the envelope gene is accessed from a spliced messenger RNA. So this 5' splice site is up here, 3' splice site is between Pol and Env. And so all of this material in the middle, GAG, Pol, is removed from the messenger RNA that is going to encode envelope. And so that's interesting, right? Because you don't want to package the envelope messenger RNA. You want to package the full-length genomic messenger RNA, the same messenger RNA that encodes GAG and Pol. So that's why that's important. In HIV and avian leukemia virus, it turns out that psi, the psi sequence, the packaging sequence, is upstream or it spans that splice site. So there has to be, and there are other mechanisms that ensure that only the unspliced messenger RNAs are packaged, right? So for HIV, the psi is necessary, but it's not sufficient. There are other sequences that we don't understand that are important to make sure that only the full-length genome and not the enveloped, or for HIV, the multiply spliced messages. Remember how many of those there are for the complex retroviruses. We only want, viruses have evolved to only package the full length. Okay, so that's really all I care that you understand about packaging the genome. I want you to understand how you define those signals and what some of the overarching issues are. And so for the next couple of minutes, I'm going to talk about acquisition of an envelope, budding, and release from cells, and that'll be the end of this session. Okay, so how do viruses that are enveloped get their envelope? Two different strategies. And the first one, the more common one here, is described using an image that shows the influenza virus life cycle. So these RNAs the genomic RNAs are being replicated in the nucleus. They're being coded by nucleoprotein that's going in. You can see that up at the top, and the M1 protein and the nuclear export protein are also getting imported, and then these ribonucleoproteins are getting exported in step number 3. They're being delivered to the plasma membrane, where the hemagglutinin and the neuraminidase and the matrix protein are being assembled. And this sort of sequential assembly of internal components here in the nucleus for influenza virus, and from the vesicular systems for the glycoproteins, is true for most enveloped viruses. So these interact. Remember, we talked about how the matrix protein helps to serve as a bridge because it interacts with nucleoprotein and the glycoproteins, and then you have budding here. The other model is illustrated by retroviruses, and for them, there's coordinated assembly of internal structures with the acquisition of the envelope. And so remember that retrovirus particles, they form by budding, but they don't mature. There's not cleavage of those internal capsid structures until after release. So here the RNA genome, which is the messenger RNA, is being exported from the nucleus and associates with the GAG polyprotein here. You can see there's the matrix part of it, the capsid part of it, the nucleocapsid part of it, and some other accessory proteins, but it's still all bound together in a polyprotein. And remember that there's a fraction, a smaller fraction of proteins that are made from that message that are GAG-Pol polyproteins. And so here, appended still on the end of that protein is the protease sequence, the reverse transcriptase, and the integrase. And all of these end up assembling at the plasma membrane in association with a glycoprotein, transmembrane and SU glycoprotein, and the RNA genome coded with some of these polyproteins also gets recruited to the plasma membrane. And so there's GAG polyprotein, there's GAG-Pol polyprotein, and there's the dimeric genome also here at the plasma membrane. And then as budding happens, and the virion is released, then there is cleavage of the polyproteins to give you the final structure. So let's talk for a minute or two about budding, which is weird, because budding is a process that has no immediate or obvious parallels in normal cell biology. So that has led virologists and cell biologists to try to understand what are the molecular signals that cause virions to bud from the cell surface. And I think what's really interesting is that sometimes there doesn't need to be, the requirements are very different. So in some cases, one needs glycoproteins and capsid proteins to interact together at the membrane. That's true in this number 1 case for alpha viruses like Synbus virus. In other cases, there are no glycoproteins needed at all, just the assembly of the matrix or capsid proteins at the plasma membrane is enough. And that's true actually for retroviruses. Budding is dependent on GAG, but you can actually get it to work experimentally without having any glycoproteins there if the matrix gets to the plasma membrane. For a virus that we haven't talked about, it's an RNA virus positive sense called coronavirus, just the glycoproteins are enough. So you don't need any of the other proteins. And then in case number 4 here in this image from your textbook, matrix proteins are important, but other proteins are important for efficiency or accuracy, and that's true for rhabdoviruses like VSV or rabies, or for orthomyxoviruses like flu. But our examples, as we've just talked about, are going to be the retrovirus case and the orthomyxovirus case, which were on the previous slide. About that, with a membrane, with having the protein-protein interactions that allowed the gag proteins to form an assemblage that was ultimately, going to have enough proteins come together so that the virion could form. And then there were sequences that were referred to as late domains, or L. And those late domains were sequences that, when altered, led to the particles basically getting stuck at the plasma membrane. And when people looked at those from across these were a lot of different people studying a lot of different viruses, it was clear that these late or budding domains had similar sequences. And so they were called late because the defect in assembly was a really late defect, about as late as you can get. And they found that there were sequences that had a lot of prolines. Here you can see PPPY or PTAP, a lot of similar sequences across really very different kinds of viruses. And people discovered that those sequences, that when they were mutated, that they could actually be moved around in the protein context and still work. So you could move them when they were mutated. The viruses tended to get stuck, as you can see in this picture. And so they proposed, because they were sort of sequence independent, that they might be docking sites for cellular factors that were involved in budding. And this is a slide from a paper from a review article on these late domains, where you can see how even in retroviruses they can be found at a variety of different places in the gag polyprotein, and also how their sequence is actually quite reminiscent of sequences that serve the same function in VSV or in Ebola virus. And really, because so much work was going on at that same time in HIV, there was so much money and so many labs that were studying HIV that it was actually pioneering studies with HIV that revealed that there were cellular proteins that identified cellular proteins that interacted with those late domains. And this table here gives you the names of some of those proteins. TSG101 was a protein that interacted with the HIV gag, and there are other proteins that were subsequently identified that interacted with these other either gag or matrix proteins that had late domains. And it turns out that those proteins are involved in vesicular budding. Those are proteins that are normally involved in vesicular trafficking from endosomes to lysosomes. And these proteins that are normally involved in vesicular trafficking were playing a role interacting with the late domains in the gag proteins and playing a role in budding. And if you look at this slide, you can think about the fact that topologically, budding out from the plasma membrane is topologically similar to budding into intracellular vesicles. So you have to think about your donut here, and what ends up being inside and what ends up being outside. And so some of these proteins have homologs in yeast and have been studied in yeast for cell division for a long time, and then in mammalian cells have been shown to play a role in sorting vesicles into other vesicles. Here's a slide from your textbook that talks about the same thing. So from this, I want you to understand, maybe be able to name the pathways that gave rise to the proteins. What are the escort proteins that are involved normally in vesicular bodies and budding into vesicles, and in cell division in yeast. But these proteins are interacting with viral proteins that help virions bud from the cell surface. So then I have the last slide, which has to do with we've talked now about how enveloped viruses bud from cells. How do non-enveloped viruses get released from cells? And that is actually still, pretty much a mystery. Couple things we know. We know that for a number of viruses, they replicate, they fill the infected cell, they can induce apoptosis or frankly just cell lysis because of the bulk, the mass inside the infected cell. So a lot of non-enveloped viruses are released by cell lysis, but actually fairly recently there are some non-lytic mechanisms that have been identified, even for viruses like poliovirus, in which viruses are actually budding into vesicles and being released in a non-lytic fashion.